we began developing the CD22 chimeric antigen receptor, quite early after the very initial reports of success with CD19 CAR T-cells. At that time, we weren't really aware of the issue with CD19 antigen loss as a mechanism of escape. But what we did know was that CD22 was a well-validated target in acute lymphoblastic leukemia and in lymphoma. We had within the Center for Cancer Research with Ira Paston had developed a CD22 targeted immunotoxin that had activity in hairy cell leukemia and ALL. And so with the success of the CD19 CAR, we decided that it would be a reasonable idea to develop a CD22 CAR as an alternative. As we learned with our initial experience in the lab, it turns out that CD22 is a more difficult target to make an active CAR. We made a lot of CD22 targeted CARs that bound well but didn't work. After a couple of years of work in the pediatric oncology branch, came up with a construct that we ultimately took in the clinic. During that entire process, it became apparent that the CD19 loss was something that we would encounter with the CD19 CAR T-cells and with blinatumomab. And so, in my mind, the importance of the CD22 CAR actually increased as we learned that was going to be a mechanism of leukemic resistance. Conceptually, in ALL, the initial development of chemotherapy for ALL, was started in the 60s with a single chemotherapeutic agent, and it became apparent pretty quickly that although there were responses to the initial chemotherapy drugs, that all of those patients relapsed. And so the concept of combining cytotoxic chemotherapy in ALL sort of was developed really in that disease, and we're now at a point where patients receive 2 to 3 years of multimodality therapy. So combinatorial treatment in ALL is an important concept already. I think that given the incidence of CD19 loss and given that we're seeing relapses in the CD22 CAR trial, a very promising response rate, but relapses with changes in CD22 site density, I think that it really suggests that combinatorial targeting approaches really make sense to see whether we can improve the likelihood of sustained remissions with chimeric antigen receptor treatment. We've certainly thought about this, in the context of gene constructs that could be used to redirect T-cell specificity, but certainly we've thought about and have talked about, other combinatorial approaches, antibody-based combinatorial approaches. The other possibility is to try to understand the mechanism of CD19 and CD22 loss, and my sense is that might be epigenetic, and there may be ways of actually combining epigenetic modifiers. Again, this is all hypothesis at this point. We have no data to that effect, but I think you can envision a number of possible scenarios to chip away at the antigen loss.